Hello students, welcome on board. This is part 4 of the chapter Sexual Reproduction in Flowering Plants. In this video, we will be covering outbreeding devices, pollen-pistil interaction, artificial hybridization, and double fertilization. For better understanding, please do watch the previous videos. Without further ado, let's begin with the first topic. The first topic is outbreeding devices. Outbreeding devices. Now, the majority of the flowers are hermaphroditic flowers. Hermaphroditic flowers, which means that they will have both male reproductive organ and female reproductive organ in the same flower. Alright. So that means hermaphroditic flowers are more likely to get self-pollinated. They are more likely to get self-pollinated as they have both male and female reproductive organs within the same flower. But continued self-pollination leads to inbreeding depression. It leads to inbreeding depression, which means that the plants will have reduced biological fitness. Plants will have reduced biological fitness. All right. So, in order to avoid inbreeding depression, the plants have developed certain devices to reduce self-pollination and to increase cross-pollination. And these devices are, first, the pollen release and the stigma receptivity is not synchronized. It's not synchronized. The next one is anther and the stigma is placed at different positions so that the pollen cannot come in contact with the stigma. All right. And the third device is self-incompatibility. This is a genetic mechanism. That prevents self-pollen from fertilizing the ovules of the same flower. All right. And the fourth device is the production of unisexual flowers. So that it increases the cross-pollination. In order to reduce the inbreeding depression, the pollen release and stigma receptivity is not synchronized. The anther and stigma are placed at different positions. The third device is self-incompatibility, which is a genetic mechanism that prevents self-pollination from fertilizing the ovules of the same flower. And the fourth one is production of unisexual flowers to increase cross-pollination. All right. Now we are going to learn how the pistil recognizes the right type of pollen grain after pollination. This is mediated by a mechanism called pollen-pistil interaction. It is called pollen-pistil interaction. Now the pistil has the ability to recognize whether the pollen is the right type or the wrong type. Now, if it is the right type, then the pistil accepts the pollen and it leads to pollen germination. It leads to pollen germination. Now, if the pollen grain is the wrong type, then the pistil rejects the pollen and the pollen germination does not take place. All right. Now, this acceptance or rejection is mediated by the chemical components which are present on the pollen grain and the pistil. Alright, the acceptance or rejection is mediated by the chemical components which are present on both the pollen grain and the pistil. Now, in this picture, you can see that the pollen grains are deposited on the stigma. Now, if the pollen grains are compatible, they germinate to form a pollen tube. Now, this pollen tube then grows through the stigma, through the style, and then finally reaches the ovary. The pollen tube enters the ovule through the micropylar end. It first enters the synergids through the filiform apparatus. The synergids actually guide the growth of the pollen tube. Now, once it reaches the ovule, it releases the male gametes into the ovule. So, now we've learned that if the pollen is compatible, the pollen germinates into a pollen tube, which grows through the stigma and style to reach the ovary, where it enters through the micropylar end, and it enters one of the synergids through the filiform apparatus, and it reaches the ovule to release two male gametes into the ovule, right? Now, the series of these events are called pollen-pistil interaction. It is called pollen-pistil interaction. So, pollen-pistil interaction is a dynamic process involving the pollen recognition followed by the acceptance of the pollen. All right. Now, we learn the process of double fertilization. Double fertilization is a characteristic of angiosperms. So, we've learned that the pollen tube enters the ovary to release two male gametes into the ovule, right? Now, one of the male gamete fuses with the egg cell to form a zygote. And the fusion of male gamete with the female gamete is called syngamy. All right. So, one male gamete fuses with the egg cell to form a zygote. And this fusion is called syngamy. It's called syngamy. All right. Now, the second male gamete enters the central cell to fuse with the polar nuclei or secondary nucleus to form triploid primary endosperm nucleus. And this is called triple fusion. All right. So, the second male gamete fuses with the polar nuclei or secondary nucleus to form a triploid primary endosperm nucleus. It forms a triploid primary endosperm nucleus. And this fusion is called triple fusion. It is called triple fusion. It's called triple fusion because it involves the fusion of two haploid polar nuclei with a male gamete. Alright. Now this primary endosperm nucleus then develops into the endosperm that provides nourishment to the developing zygote. Alright. So we've learned that in double fertilization, one of the male gametes fuses with the egg cell to form a zygote, and this fusion is called syngamy. And the second male gamete fuses with the polar nuclei or secondary nucleus to form a triploid primary endosperm nucleus. And this fusion is called triple fusion. Then later, the primary endosperm nucleus develops into an endosperm that provides nourishment to the zygote. Since there are two fusions taking place in an ovule, that is syngamy and triple fusion during fertilization, this process is called double fertilization. Alright. Now the next topic is artificial hybridization. Artificial hybridization. Now in artificial hybridization, desired pollen grains are used for pollination, and the stigma is protected from the contamination by unwanted pollen. Alright. So in artificial hybridization, the desired pollen grains are used for pollination and the stigma is protected from the contamination by unwanted pollen. Alright. And this is done by two procedures called emasculation and bagging. It is called emasculation and bagging. Now in emasculation, anther of a bisexual flower is removed from the bud using forceps. The anther of a bisexual flower is removed from the bud using forceps, and this is done before the anther dehisces. Alright. Now this emasculated flower is covered with a bag to prevent contamination of the stigma by unwanted pollen. And this procedure is called bagging. It's called bagging. So in emasculation, the anther is removed from the bud using forceps before the anther dehisces, and then the emasculated flower is then covered with a bag to prevent the contamination of stigma by unwanted pollen, and this is called bagging. Alright. So that's it for the session. This was part 4, where we've learned about outbreeding devices, pollen-pistil interaction, double fertilization, and artificial hybridization. In part 5, we'll be covering post-fertilization structures and events. If you found this helpful, please do like the video, and if you have any doubts, comment down below. Subscribe to watch the upcoming videos. Thank you for watching.